Bismillahirrahmanirrahim. Paper C, Cytology, Genetics and Evolution. From the portion Cytology, according to syllabus, Chapter C, Chromosome Morphology and Karyotype Analysis. Chromatin, a dense material that is consist of DNA and protein. It appears as a thread-like structure during cell division known as the chromosome. Chromosomes are considered as the physical basis of heredity. The small bodies located on the chromosome are known as genes. Genes are arranged on the chromosome like beads on the string. Genes are the unit of heredity that transmit the characters from one generation to the other. Chromosomes were discovered by Wilder in 1876. After that, Sutton in 1908 identified the function of chromosome. Structure of chromosome. Structurally, the chromosome consists of following parts: chromatids, chromonema and chromomeres, centromere or primary construction, nucleolus organizer or the secondary construction, and telomere. Diagram showing different parts. This is the thread-like structure which is either known as the arms or the chromatids. Two arms are connected by means of central part that is known as the centromere. The chromatid consists of this highly coiled thin filaments that is known as the chromonema or chromonemata. On the chromonema or chromonemata, these bead-like swellings are present that is known as the chromomere. The region between two chromomere is known as the interchromomere. The outer covering of the chromatid or membrane is known as the pellicle. Inside the pellicle, the central part present is known as the matrix, and in the matrix, this chromonemata or chromonema are present. This is the centromere or the primary construction, also known as the kinetochore, with the help of which chromatids are attached to one another. The centromere, during the cell division, they unite with the microtubule, so help in the formation of spindle fiber. This help in the chromosome movement. कुछ क्रोमोसोम में हमारे पास ये एडिशनल पार्ट बनता है. इट इज़ नोन एज द सेकेंडरी कंस्ट्रक्शन as it is associated with the nucleolus, therefore it is known as nucleolus organizer. Secondary construction के बाद chromosome का short सा part present है, it is known as the satellite, and this is the end part or the polar region that is known as the telomere, the function of which is it prevent the attachment of other chromosome. Types of chromosome on the basis of centromere number: acentric, the chromosome without centromere; monocentric, having one centromere; dicentric, chromosome having two centromere; polycentric, chromosome having more than two centromere. Second, on the basis of centromere position: telocentric, it is of rod shape when centromere is present at the proximal end; acrocentric, rod shape centromere between two chromatids, one arm is of very short length, other is long. Submetacentric, J or L shaped centromere in the center, one arm short, other long, means two arms are of unequal length. Metacentric, V shaped centromere is in the center, two arms of almost equal length. Diagram showing different types of chromosome on the basis of position of centromere. First is the telocentric, centromere at the proximal end; acrocentric, centromere between two chromatids, one arm is very short, other is long. Submetacentric, centromere between two chromatids, two arms are of unequal length. Metacentric, centromere is in between the two chromatids, two arms are of almost equal length. Molecular structure of the chromosome: it consists of DNA and protein. There are two distinct regions of the chromosome: euchromatin and the heterochromatin. Euchromatin, it is of light color, it is larger part and it is active, helps in the formation of RNA. Heterochromatin, it is of dark color, it is inactive and it do not take part in the formation of RNA. Kinds of chromosome on the basis of function: two types, autosome and sex chromosome. All the chromosome other than the sex chromosome are known as the autosome, and sex chromosome that determine the sex, either X or Y chromosome. And there is special type of chromosome that is Lamb-Rush, Polytene and B chromosome. First one is the Lamb-Rush chromosome. It is found in the spermatocyte nuclei of Drosophila and the oocyte nuclei of both vertebrate and invertebrate. It was discovered by Ruchard in 1892. It is of very large size, consists of this central axis to which this chromosome or the DNA fiber is attached. The DNA fiber surrounded by the matrix that consists of RNA and proteins. This DNA fiber, it form the lateral loops. ये हमारे पास loops हैं. At the time of cell division, the number of loops increases, and during the process of meiosis, cell division, the number of loops increases, and in the diploidine stage, the number of loops may reaches at their maximum. यानी जब डिप्लोटीन स्टेज आती है तो जो नंबर ऑफ लूप्स हैं उनका नंबर सबसे ज़्यादा हो जाता है. लेकिन जैसे जैसे मियोसिस फर्दर प्रोसीड होती है, फर्दर स्टेप्स आते जाते हैं, द नंबर ऑफ लूप्स जो हैं वो डिक्रीज होते जाते हैं एंड फाइनली दीज लूप्स डिसअपीयर. Second is the polytene or salivary gland chromosome found in the salivary gland of the Drosophila. They were discovered by Balbiani in 1881. It is of very large size, may reach up to 200 times more than the normal chromosome. इस क्रोमोसोम में हमारे पास अल्टरनेटली डार्क एंड लाइट बैंड प्रेजेंट होते हैं. डार्क बैंड हमारे पास यूक्रोमेटिन है एंड लाइट बैंड विच इज़ ऑल्सो नोन एज द इंटर बैंड, इट इज़ heterochromatiन. As the light band is present between two dark bands, therefore it is also known as the interband. And last type is the B chromosome. It is different from the autosome and the sex chromosome because they are without any genetic function. Therefore, they are also known as the accessory or supernumerary chromosome. They are found in the maize, some insects, and small mammals. In some organisms, the B chromosome consists of heterochromatin like maize and insects, while in some organisms they are consist of euchromatin, for example rye. Karyotype analysis: when chromosome of a species is arranged according to size, shape, structure, length and the position of chromosome, this is known as the karyotype analysis. And when the chromosome is shown by a figure, this is known as ideogram. Ultrastructure of the DNA ultrastructure of chromosome. Chemically, the chromosome consists of nucleic acid, DNA and RNA. Proteins, they are of two types: histone or the basic protein, non-histone or acidic protein, and the metallic coins. Nucleic acid, it is present in the form of DNA and very small amount of RNA is present that is responsible for the protein synthesis. The DNA is a double helical structure that consists of nucleotides. In the electron microscope, the DNA look like a string of beads, and these beads or the knob-like structure, it is known as the nucleosome. Proteins are of two types: histone or basic, non-histone or acidic. Histone protein are of five different types: H2A, H2B, H3, H4 and H1. The non-histone or the acidic protein, they have regulatory and the enzymatic function. They control the physical properties of genes, for example phosphoprotein, RNA polymerase and DNA polymerase. And the metallic ion present are the magnesium and calcium. They make the organization of chromosome. This is the structure of nucleosome. H2A, H2B, H3 and 4. ये हमारे पास टोटल इनका नंबर 8 होता है. therefore it is also known as the octamer. This is surrounded by means of this DNA helix and it is linked by means of another protein that is H1. This whole structure is known as the nucleosome. जो न्यूक्लियोसोम में हमारे पास प्रोटीन है उनमें ऐसे अमाइनो एसिड प्रेजेंट होते हैं जिन पे हमारे पास पॉजिटिव चार्ज होता है. देन द सोलेनॉइड मॉडल, ये हमारे पास डबल स्ट्रैंड डीएनए है. डीएनए जब हिस्टोन प्रोटीन के साथ मिलते हैं तो हमारे पास स्ट्रक्चर बनता है न्यूक्लियोसोम. दिस न्यूक्लियोसोम दे फॉर्म अ फाइबर दैट इज नोन एज द न्यूक्लियोफिलामेंट. द न्यूक्लियोफिलामेंट इज फर्दर coiled up. This is the further coiling, as a result of which a supranucleosomal structure is formed that is known as the solenoid. The supranucleosomal structure or the solenoid, it consists of these 6 nucleosomes. Means in one turn of the solenoid coil there are about 6 nucleosomes. And then this is the scaffold loop. These are the non-histone protein that play a role in organization and the compaction of the chromosome. And this is the structure of complete chromosome, centromere and chromatid or arms.